All right, now it's time to talk about making a protein. So we've got our functional mRNA, and now it's time to translate the mRNA into protein. So making the mRNA was transcription. Translation is turning the mRNA into protein. And so we have three main players involved here. The first is mRNA, which is the recipe for our protein. The second is the ribosome, which is the stew pot where we're going to make our recipe. And then tRNA is what puts the ingredients in the pot. And of course, the ingredients are amino acids, because a protein is made of chains of amino acids. So mRNA is the recipe for making a protein. The ribosome is the site where the protein is made. tRNA is putting amino acids into the growing protein. So let's take a look at this, shall we? Let's look at the ribosomal function. Again, the ribosome is the site of protein synthesis. And the details here are that the ribosome is where mRNA instructions are used to bring specific amino acids to a growing chain. And the amino acids at the ribosome bind together to make that growing chain. So it's where mRNA meets up with tRNA to bring some amino acids, and the amino acids bind together. So again, I mentioned tRNA because the ribosome doesn't have a large supply of amino acids just hanging out next to it. You need tRNA, transfer RNA, to bring amino acids to the ribosome. So transfer RNA is a big hunk of RNA attached to a specific amino acid. In fact, for every amino acid, there is a specific tRNA. And furthermore, there can be different tRNAs attached to the same amino acid. So you might have the amino acid arginine, and you could have, tRNA A code for, attached to an arginine, and tRNA B could be attached to an arginine, and tRNA C could be attached to an arginine. And we'll see how that works as we continue in the chapter. But for now, tRNA is attached to an amino acid. And that tRNA binds to the mRNA at the ribosome, where it brings, and so its whole job is to bring amino acids to the ribosome so the chain can be produced. So when we talk about bringing specific amino acids to the ribosome, we have to have a way to code for specific amino acids. And it has to be better than just the gene codes for a protein, because a protein is made up of multiple different kinds of amino acids chained together and then folded and twisted together. Well, the way we know which amino acid to use in a specific part of the chain is through the fact that mRNA is made of codons. What does that mean? What is a codon, you ask? Well, it's very simple, actually. A codon is just a set of 3 nucleotides. 3 nucleotides is 1 codon. So codons are made of nucleotides, the A, U, G, and C. Each codon codes for a single amino acid. Page 180, I believe, of your book has a table with the codons and what amino acids they correspond to. But let's take a look at this. AUG is a codon you're going to need to know by memory, because AUG codes for start. In other words, in order to actually begin making a protein, you have to have the AUG codon, because the AUG codon says start here. So where the ribosome first binds to the mRNA is at the start codon. And then we keep reading codons on and on and on throughout the entire length of the mRNA until we reach the final codon in that mRNA, which is a stop codon, which tells it to be done. There are 3 different stop codons, UAA, UAG, and UGA. It would be good to know at least one of those. So here's a nice little picture I have. So any one of these is a nitrogenous base, UGCA. These are all the nitrogenous bases you find in mRNA. A codon is a set of 3. So here's CGG. And the codon CGG in mRNA codiert voor aminozuur, arginine. Whereas the codon ACC in mRNA codiert voor aminozuur, threonine. Obviously, I could not find a picture in English that was this nice. So here's Dutch, in which the codon CGG in mRNA encodes for the amino acid arginine or arginine. And ACC encodes the amino acid threonine, THR. You're not going to need to memorize what codon codes for what amino acid, because there's a lot of codons in here. In fact, let's look at a table of the different possible codons you see. And this isn't even all the codons. This is the 20 codons, or these are codons for the 20 most common amino acids seen in humans. So here we go. UUU codes for the amino acid PHE, phenylalanine. And funnily enough, UUC also codes for phenylalanine. UUA codes for leucine, as well as UUG. CUU, leucine. All 4 of these code for leucine. So here's AUG. Remember, that's our start codon. So they made it green because you start moving at the green light. What's interesting about AUG being the start codon is that it codes for the amino acid methionine. So every single protein there is in life starts with methionine. Kind of neat. And so you got all of these codons here. There's an easy way to find out what your particular codon codes for. Say you've got CCG. I want to know what that is right offhand. Well, the first letter in that codon is C. The second letter is C. And then I just find CCG. So the first letter tells you what row to look in. The second letter tells you what column to look in. And once you get to the right box with the row and the column, you just find the third letter in the codon. CCG codes for proline. So you don't need to know all the names. I know a lot of these names just because of experience. But obviously, you don't need to know that this is tyrosine, that that's proline, phenylalanine, leucine, isoleucine, valine, alanine. Yeah, you don't need to know all of those. So you're just concerned with the 3-letter abbreviation for the amino acid and knowing how to read this table. Because, surprise, surprise, this table will be part of your exam. mRNA arrives at the ribosome. And the first codon read is the start codon. AUG, so the first amino acid to arrive is methionine. All protein synthesis starts with methionine. So our ribosome needs a methionine. We need to get a methionine over to this ribosome. So we have a tRNA that has a methionine. How do we get the tRNA to bind to the mRNA so it can bring the methionine down? Well, it's very simple. Because tRNA is made of RNA. And mRNA is made of RNA. So each of them has nucleotides. And those nucleotides have nitrogenous bases. And nitrogenous bases like to pair up in specific ways. In RNA, adenine likes to pair up with uracil, guanine to cytosine. So the RNA codon will bind to the proper base pairs on the tRNA. And so we call that set of 3 nucleotides on the tRNA that can bind to the mRNA codon the anticodon. So tRNA has an anticodon. And that's the tRNA binding site to attach to mRNA. Anticodon is, again, base pairs to the mRNA codon. So if the mRNA codon for start is AUG. What's the tRNA anticodon? Oh, I think I'm hearing it. Best speak up. Come on. Come on. A, what's the complement to A? It's the base pair. Yeah, that's right, U. Okay, the next one in the codon is U. What's the complement there? Yep, very easy. A, and then we have a G, which has the base pair C. Right. So if our mRNA start codon is AUG, the tRNA carrying methionine will have an anticodon of UAC. And that way, UAC can bind to AUG, and we develop a nice strong bond, allowing our methionine to come to the right place at the ribosome. Very easy. Just a little bit of stuff to memorize. So, protein polypeptide synthesis. So I'll use the word polypeptide interchangeably with protein because a polypeptide is a chain of amino acids, whereas a protein is when you take that chain and start folding it up and twirling it up and compacting it into a specific ribbon shape that's active. So we're primarily concerned with the making of the chain, not so much the protein getting its primary, secondary, tertiary, and quaternary structure. So polypeptide synthesis begins with the start codon, AUG. And the tRNA with its UAC anticodon comes along and binds to the start codon, and it brings methionine. Now we need the next tRNA. Well, how do we keep it all going smoothly? We're at the ribosome, and it is the ribosome's job to keep things going. So the ribosome has binding sites for tRNA. It has 3 binding sites. The first we call the A site. I like to call it the awaiting site, where a tRNA is awaiting to become part of the polypeptide chain. So it's waiting for its amino acid to be added to the chain. The next site is the P site, which I call the processing site, where we take the tRNA at the P site and add its amino acids to the chain. So, bam, adds its amino acid. And then finally the E site, or the exiting site. So this is where a tRNA that has already added its amino acid to the chain and is now empty takes off. It's released from my mRNA. So an empty tRNA is released from the mRNA in the ribosome. And this just continues ad infinitum over and over and over and over again until you reach a stop codon. So here we go. This is a tRNA at the P site of a ribosome. And here's the tRNA anticodon combined with the mRNA codon. So here we go. Very easy. This is our mRNA at the ribosome. This is our tRNA at the ribosome bound to the mRNA. And this tRNA is carrying its specific amino acid. So let's look at some videos. I've got a few here, and we'll just go through them. I'll help you out with what's useful information and what you don't need to know. So without further ado,
translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has 2 binding sites for transfer RNA, the P and A sites.
Now, this movie doesn't have the E site in it, unfortunately. You do need to know the E site. So in this one, they're going to show tRNA sort of leaving the P site and then popping off free over here. So where it pops off there would actually be the E site. Stupid dissonance in biology.
Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet.
So here we go. This is our basic setup. This guy is going to be processed, and this guy is awaiting the addition of an amino acid.
Amino acid carried by the second transfer RNA binds to the methionine.
Bam.
The first transfer RNA leaves the P site, and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached.
Okay, here's our stop
codon. A release
codon.
factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
Boop, that easy. All right, let's look at another video because I want to be able to just drive this home to you guys.
Translation.
Alright. YouTube doesn't want to work, apparently. Well, go back. This third one will work.
mRNA, the substrate for translation, has a unique 3' end called the poly A tail.
There's that poly A tail with all those adenines.
mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end.
The 5'
Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds.
So here they're using initiation site. That's just start codon. So you don't need to know the term initiation site.
First mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
Yeah, you don't need to know that P stands for peptidyl and A stands for aminoacyl. Just P and A site. And again, it's ignoring the E site.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
So this is elongating the chain. And it just
These are the
continues
basic
on and
steps
on.
of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, The ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
That was easy. Now the problem is I want this first video because it was good. So we're going to have to just do this because YouTube is being a jerk, or PowerPoint is being a jerk. One of them. I don't know who's being a jerk. Let's try this out here. Here we go. So this video is going to be all sorts of complex. Ugh, really? Ray advertisements. Skip. 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 Skip, 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 skip. There we go. So this video is going to be a little bit complex. Let's find it here. Poly A tail, methylated cat. So that's where tRNA is binding. And then here we have our A, P, and E site. So we already know that the ribosome assembles around the AUG. And then tRNA binds to the AUG, and that's the tRNA carrying UAC. So, because that's UAC is the anticodon to AUG, and it has methionine. And then the next tRNA comes along at the awaiting site, the A site. And so the amino acid being processed at the P site is going to be added to the amino acid at the A site. So let's just look at it now that we have our full APE. Yeah, this one has no sound, so I guess I have to talk uncomfortably through it. So it's transferred there, and then we get to the E site. Our next tRNA comes along, amino acid number 3 here. That original tRNA exits and we transfer. The process just continues until we get to our stop codon where a release factor arrives and the whole thing falls apart. There we go. That's our polypeptide. So, hopefully these videos will sort of help it make a bit more sense for you. And let's review the stop codon real quick. Stop codons are how we end translation. So tRNA does not in fact bind to a stop codon. Instead, we have a release factor. And it binds to the stop codon at the A site. And it adds a water. It carries a water which hydrolyzes the bond between the tRNA at the P site and the growing amino acid chain. And that releases the whole amino acid chain. Hydrolysis releases the polypeptide chain to go along. So there are a few ways transcription and translation can be messed with, and that's gene mutations. Now, gene mutations occur in the DNA, in the gene. And if you alter DNA, you alter mRNA. And if you alter mRNA, you alter the protein. So gene mutations cause alterations in gene expression or alterations in protein synthesis. There are a couple of different kinds of gene mutations. One is the point mutation. In the point mutation, you have a single base pair changed. So maybe it was originally an A to T, and suddenly it's changed to a G to C. That's a point mutation, a single pair altered. Point mutations can result in a number of different things. The grand majority of these are negative or harmless. If it's harmless, in general, it is classified as a silent mutation. A silent mutation still codes for the same amino acid. In other words, even though we changed the base pair being read there, and so we end up changing the nucleotide in the mRNA, it still produces a codon that codes for the same amino acid as before. So we can take a brief look here at our codons. So, say your DNA originally had AAT. So AAT on the DNA. That would be transcribed into UUA. But say a point mutation occurs, and instead of AAT, it becomes AAC, which is transcribed into UUG, but both of them still code for leucine. So it's a silent mutation. It doesn't actually cause any kind of changes. Then there's the missense mutation, and that alters the amino acid produced. So a missense mutation changes the amino acid in the polypeptide chain. And this could be very trivial, or it could be dangerous. In general, missense mutations aren't really classified as all that dangerous, because changing a single amino acid in a protein that's thousands of amino acids long is not going to do a whole lot. However, there are certain areas on a protein that are vital to its functioning. For instance, an enzyme has that active site. That active site has a specific shape. If you get an alteration of even a single amino acid, it may alter the shape of the active site on the enzyme, and that may screw up the whole enzyme. So, unless a crucial area or active site is altered in a missense mutation, neither a silent or a missense mutation is going to do much damage. However, there is the nonsense mutation. The nonsense mutation is when you completely screw up the resulting protein. It could be something as terrible as altering the active site. Even a single amino acid alteration in the active site could destroy the protein. The enzyme might not function at all. Or, if we take a look at our codons here, say you have, where is it? We have UAU. Or no. Let's go with UAC. There we go. So we have UAC as a codon, right? Codes for tyrosine. The original gene that produced this codon would be ATG. So ATG. Now say there's a point mutation and our ATG becomes ATC. If it's ATC, the resulting codon on the mRNA, A to U, T to A, C to G. Instead of coding for tyrosine, we now have a stop where there shouldn't be a stop. So a nonsense mutation could alter a crucial area of a protein and make it non-functional, or it can create a stop codon where there shouldn't be one. So these are all bad. But there's another kind of mutation that's even worse, and that's the reading frame mutation. A reading frame mutation consists of either insertions, in which you add one or more nucleotide pairs to a gene, or a deletion, in which you delete one or more nucleotide pairs. So, this is very bad, because our gene is translated into mRNA, and that mRNA is read as sets of 3. So ATG to MET, because ATG becomes UAC, or it becomes AU. Well, wow. Whoever made this image actually screwed up the whole fact that there's no thymine in RNA. Oh, shame on them. So AUG codes for methionine. GAA codes for glutamine. GCA codes for alanine. CGT codes for glycine. But if you delete my G here, it becomes AUG, AAG, CAC, GU. So we delete this nucleotide and move the frame over. And it screws up absolutely everything after the deletion. Everything after the deletion is screwed up completely. So we have AUG making methionine as normal, but suddenly we have lysine instead of glutamine and histidine instead of alanine. So it's all screwed up. Here, GU, that doesn't code for anything. That's only 2 amino acids. That's not an intact codon. So everything's screwed up. Frame shifts just almost universally result in a very negative impact on an organism. So luckily, cells have mechanisms in place to try and counteract mutations. You can often edit their DNA if their mutation occurs and reset it back to normal. By the way, this is a deletion here. An insertion does the same kind of frame shifting, but instead of like removing this G, we might add a C. And so it would be AUG, CGA, AGC, ACGU. So you can see how an insertion or a deletion would totally mess things up. So the central dogma of biology is gene to RNA to protein, no matter the cell. This is one of those universal things. If you are a cellular life form, you take DNA in the form of genes, make mRNA copies of them, and use the mRNA to produce protein. This is one of the strongest pieces of evidence for an evolutionary link between all life forms, because it is a shared system. The earliest form of cellular life must have shown DNA to RNA to protein, because all life since has shown DNA to RNA to protein. So everything evolved with this system. So this is truly a link between all cellular life forms. Between this and the fact that all cellular life forms undergo glycolysis, we can see pretty clearly that all life on Earth is related through evolution. And brother, that is awesome. To think about how all cellular life is a family. Pretty neat. So we call it the central dogma of biology. All right, that's it for this. We'll pick up with the rest of Chapter 9, talking about viruses and how scary they are, and how really all you should be doing is curling up into a fetal position in a corner, shaking and saying, I'm going to die, I'm going to die, I'm going to die.